Hi, so my name's Linda. Thank you for that introduction. And today I'm going to be talking about assigning taxonomy to microbiome viruses. So a bit about my timeline. Maybe you're wondering how I know Python. Is she corrupting her Conda environment, or is she writing unit tests before she even writes any code? So I would say I am somewhere in between. I did a conversion computer science degree in UCD. To say I went from zero to hero would be a bit kind, but I think I'm somewhere middling. And I worked as a data analyst after that, and then I kind of got really interested in the microbiome, and I had a microbiome or microbiological awakening, because I was interested in other parts of biology before, but this is the first time I really became interested in microbes. So I thought, how can I get into bioinformatics? How can I use Python to ask and to address these biological questions. So I joined UCC as a research assistant in their APC microbiome department, and this is where I first cut my teeth with bioinformatics data, and now I am a PhD student in the APC microbiome. So I'm very aware that this talk is my area of research is very tangentially related to healthcare. So if you have this umbrella of healthcare, you have bioinformatics, then you have microbiome analysis, which I'm not really sure in the mainstream if that's microbiome analysis is used as clinical indicator, and then I'm studying the virome aspect of the microbiome. So bacteriophage taxonomy and virus discovery I'm not sure how related it is to healthcare at the moment, but these viruses, so this here is a crassphage virus, and that was discovered completely computationally using metagenomics data, and that was only discovered in 2014. And that's the most abundant virus in the gut. It's found in 73% of humans in remote populations all over the world, so it's a really cosmopolitan virus. And there's kind of like an ancient molecular arms race going on between bacteria and viruses, and I'll spare you the details, but there's some really elegant and sophisticated genetic battles happening between viruses and bacteria, and by studying this we learned CRISPR, that CRISPR is actually a bacterial defense mechanism against these guys. So potentially in the future we can, through virus discovery, maybe it would be related to healthcare if we can mine novel proteins with different functions. So we're in this age of metagenomics. Some researchers call it a revolution in metagenomics and metatranscriptomics, because we're generating all this data from the environment. So what is metagenomics? You can go out, you can take a sample from soil, from anywhere in your environment, you can extract the DNA from there, and then you are loading it onto your computer, and then all of the computer analysis follows suit with this. And this is where Python comes in. So Python is at the moment having a huge role in virus discovery through machine learning, even identifying that the sequence is viral in the first place, checking the quality of your sequences, and then protein structure prediction is huge at the moment in the field of bioinformatics. So yeah, I think I already talked about what crassphage are, this character in the human gut, and there's estimated to be millions of viral species, but at the moment there's only 13,000 viral species that are recognized, and this is what we call in the field viral dark matter. So you have these sequences in usually when you have your metagenomic data, the first thing you would do is you would align your sequences against reference databases to see, do you get a hit? Is there anything known in your sequence? But we have all these viral sequences which we have nothing else to compare them out to in the world, so the number of viral sequences and viral diversity in the biosphere is huge. So a typical virome analysis pipeline, you would use metaspades, that's for assembly, so your DNA is fragmented into little bits. There's a Python wrapper around metaspades. I originally thought it was a Python program, but it's written in C. And then it just iterates across your DNA sequences, and like a puzzle, it assembles them, the longest ones, like reconstructing a puzzle, and then you get your assembled sequences or genomes. Viral sequence identification. Vibrant is a very good machine learning approach. VirSorter 2 is actually really popular in the field, but that's in Perl, I think. How does Vibrant work? So the essence of these programs is what they're doing is they're collecting viral sequences, diverse ones from different families, and then you put it through your scikit-learn neural network, and then it trains to learn the features associated with these different viral families. And then if you have unknown sequences, you can pass them in to your program. Can it recognize, is there some pattern in the data that's similar? And then we can say, okay, this virus is this kind of virus. CheckV, this is a Python program that's used to check the quality of your sequence. Is it complete? Is it fragmented? Is it 90% complete? Protein structure prediction. So a lot of these proteins are unknown. What can you do about it if you have found your proteins? AlphaFold is a protein prediction thing. It's 2 terabytes of data that the database is used. So yeah, the barrier to entry in bioinformatics is quite intense. Like, I think for Metaspades, assembly requires 250 gigs of RAM at some stage. So I think that like rules out a lot of average laptop and PC users. The databases involved are huge. Just a couple of days ago, Facebook, so even Facebook are in on this protein folding. They released this evolutionary scale modeling metagenomic atlas. It's a 20 terabyte database of 617 million protein structures. Okay, and then taxonomy, the part that I'm interested in. We have some programs which are existing, but I will explain why I want to do it in a different way. vContact, it clusters the sequences, and based on shared proteins, it just clusters the viruses together and into different groups, but it doesn't really tell you at a granular level what species it belongs to, or what family of. I think it can go down to family level. Pyani is also a Python program which it can compare at the nucleotide level the sequence similarity between 2 sequences at a nucleotide level. So what are the challenges of assigning viral taxonomy? There's no universal marker gene between all viruses. So in bacteria, you have this 16S rRNA gene, which is highly conserved. So you can trace that back to the last universal common ancestor. But it's thought that viruses evolved independently over around 7 times. So there's not, you can't just look at one gene and compare how they diverge. They're lots of separate groups. Evolution is not linear. Viral genetics is really funky. They can swap out and change genetic modules and genes. Also, there's no unifying method or principle in assigning taxonomy to viruses. So there's this international committee on the taxonomy of viruses, and there's researchers around the world, and they have kind of like viral study groups. So they would study crassphage, or they would study gubaphages, or different types of viruses. And for assigning taxonomy to them, they might use a different conserved gene that is unique to that type of virus. So to try and find relatedness between all viruses is a bit of a challenge, but that's what I've been assigned to as my task for the PhD, to build a tool which can compare the relatedness of all existing microbiome viruses. So first, you have your viral sequence. You need to find the proteins, and this always continues to blow my mind how the code of life it lends itself so well to be looked at and explored using our coding language. So how can you find where the genes are? You can iterate across your sequence looking for your start codons that signifies the start of a gene that will be translated, and different organisms have different translation tables to add layers of complexity onto this. But once you find that your gene, which will code for a protein, this is called an open reading frame, so it's the sequence between your start and stop. In bacterial genomes, 2/3 are known, 1/3 of these proteins are unknown. So you can predict that it's protein, but you don't know what it is. And this relates back to these known unknowns. I don't know how many, what the ratio of this in viral sequences are. So what we are trying to implement is this concept of symmetrical best hits. So I don't know if anyone is familiar with BLAST. BLAST is a sequence aligner. So you have your virus proteome A here, and this first blue segment is a protein, and so there's 4 proteins in this whole proteome, and there's 3 in virus B. So how can we compare this concept of symmetrical best hits is comparing the relatedness between these proteins. So you have your BLAST, which aligns 2 proteins together. So you call your BLAST program. The first thing it will do is it will try to align that first protein in proteome A against each protein in proteome B. This is to try and get an alignment hit. So then it will essentially interact with try to find an alignment between all proteins in each proteome. And then you get an output something like this, which tells you, okay, protein A aligned against protein B, the alignment length was 193 amino acids, and you get an e-value, a confidence score, and stuff like that. So to go more into this symmetrical best hit, so what makes a protein, 2 proteins from 2 different viral genomes symmetrical best hits is if you blast one back and it gives you the other one. So to try and explain this visually, protein A aligns against protein B here, and there's 300 amino acids where it's the query protein is aligning over the target protein. And then we have this metric where you're filtering by coverage. So, okay, the total length of the alignment from query A against the target protein B is 300 divided by the total length of the protein B in virus B, and then get the percentage. That's 62.5% coverage. So I'm filtering all of the hits by 50% coverage is the cutoff point. Now, if you blast protein B against protein A, will it also have a coverage of 15% against protein A? So this is what defines a symmetrical best hit. So what I'm trying to do is to find these symmetrical best hits between pairs of proteins in all viruses. So I can calculate these symbet distances between these viruses. This is fine. I can use this distance formula to create this matrix, and I can plot these trees. So this is quite intense. I'm trying to do this for tens of thousands of viruses. So I'm working on it at the moment to scale it up, because not all programs are made equal. Maybe I can run this and it might run for days, but I'm trying to do it more efficiently. So yes, so like if I'm blasting 10,000 viruses against 10,000 other viruses, that's 100 million operations. So what I was thinking of doing is to really redesign my program and my architecture and try to make it as most efficient as possible. So I will try this multiprocessing step where I will concurrently blast the 2 protein sequences against each other, because at the moment everything is just running synchronously, and it's going to be time consuming, and I want to try to scale this up to accommodate hundreds of thousands of viruses. I was telling some about my problem. They said maybe to use PySpark. If anybody has any recommendations of database tools or any thoughts, I would appreciate that. So this is really what I'm working on at the moment using Python. So I don't know, maybe I will write it in Bash instead, and then from inside Python call Bash. But what I'm hoping to do is I'm going to have build this tree. I'm going to plot this tree of viral relatedness based on symmetrical best hits between viruses, and then the tool in the end should I input my unknown viral sequences, and then it's going to iterate in a smart way around the tree, and then blast against sequences, and then place where the virus should be on the tree, related in terms of where it's related. Yes, so that is me. Really, I wrote a peak procrastination. I made a bot which scrapes research and puts it on Twitter. So I told my lab about this, and they were like, "Oh, phage papers. There's already one that does that." So okay, thanks. So yeah, that's it really from me.